Aligned electrospun fibers direct the growth of neurons in vitro and are a potential component of nerve regeneration scaffolds. Here, electrospun fibrous scaffolds are generated, and primary rat sensory and motor neurons are grown on them. First, fibrous scaffolds are electrospun onto glass cover slips. Primary neurons are then obtained from embryonic day 15 rats and cultured on the electrospun fiber scaffolds. The neurons are allowed to adhere and grow. Then, they are fixed and stained using immunocytochemical techniques. Imaging results reveal changes in the morphology of neurons cultured on electrospun fibers, indicating that the nanotopography of the electrospun fibers can direct neurite outgrowth.
The main advantage of this technique over existing methods, such as the use of transformed cell lines, is that primary neurons in vitro more closely mimic the behavior of neurons in vivo than do transformed cells.
These techniques offer viewers the ability to use electrospun fibers with other cell types. Alternatively, viewers may wish to use cultured motor and sensory neurons for other investigative purposes.
Generally, individuals new to this method will struggle because electrospinning is a finicky process, which we explain in more detail in our accompanying video article.
To prepare electrospun fiber substrates, begin by making an 8% solution of 85 to 15 polylactic co-glycolic acid, or PLGA, in chloroform, and a 4% Poly-L